Good morning and good afternoon, everyone. This is Mahalea Marshall with the IQ Consortium Secretariat. I'd like to thank you for joining today's webinar entitled Immunogenicity Risk Assessment, Insight into Industry Practice. This webinar is presented by the IQ Immunogenicity Working Group and is sponsored by the Clinical Pharmacology Leadership Group. I'd like to introduce today's speakers. Our first speaker and moderator today is Christine Grimaldi. Christine is an immunologist by training. She has 17 years industry experience in a broad range of modalities across discovery, preclinical, and clinical stages. She currently leads the assay development group in the Bioanalytical Sciences Department at Regeneron. Our next speaker is Kelly Coble. Kelly is a biologist by training with 27 years industry experience in regulatory bioanalysis supporting small and large molecules at preclinical and clinical stages of development. She currently leads the Global Bioanalysis Group at Boehringer Ingelheim. And finally, our last speaker, Shelley Beluski. Shelley is a board-certified toxicologist with a PhD in immunology. She has over 20 years of industry experience and is currently the head of bioanalytical development at Xencor, where she leads a team that is responsible for the innovation and implementation of custom assays supporting PK, ADA, biomarker, and potency endpoints through protein therapeutic drug development. And with that, it is my pleasure to turn things over to our first speaker and moderator, Christine Grimaldi. Christine?
Hi everyone. Today it is my great pleasure to welcome you to the second webinar presented by the IQ Immunogenicity Group entitled Insights into Industry Immunogenicity Risk Assessment Practices. For those of you not familiar with IQ is the International Consortium for Innovation and Quality in Pharmaceutical Development. It was established in 2010 as a technically focused non-for-profit organization comprised of 50 pharmaceutical and biotechnology companies. First, the vision is to be leading science-based organizations advancing innovative solutions to biomedical problems and enabling pharmaceutical companies to bring quality medicines to patients. And the mission, as a technically focused organization of pharmaceutical and biotechnology companies, IQ advances science and technology to augment the capability of member companies to bring transformative solutions that benefit patients, regulators, and the broader R&D community. Today I'm going to kick off the presentation of the survey that the IRA subgroup of the IQ Immunogenicity Working Group conducted in 2022 to gain insight into IRA practices. The results and recommendation from this survey were published in CPT earlier in 2025. And I'd like to acknowledge the great efforts by our team, which was made up of the companies shown here, and were instrumental in writing their survey, interpreting the survey responses, and writing the manuscript. Before we dive into the presentation, I would like to give you some background on the Immunogenicity Working Group, which formed several years ago. We started off as a larger group, which split into two teams. One team to evaluate the IRA practices, which is what we're going over today, and the other team which evaluated ADA data analysis practices, which presented their webinar a few months ago. Some of the topics that we're going to talk about today include the information we compiled on the industry practices for IRAs and ISIs, insight into the application of predictive tools and preclinical approaches that inform the IRA, and how the IRA informs the bioanalytical strategy. Here's our agenda for today. We're going to talk about the survey overview and summary, key learnings and highlights, recommendations, and then we'll conclude with a live panel Q&A session. I'd like to point out that we're not going to go through all of the questions in this survey, but if you're interested, you can see the questions and the responses in our publication. The speakers for today are myself, Christine Grimaldi, and I am a director in the Bioanalytical Sciences Group at Regeneron. Kelly Coble, who is Global Head of Bioanalytical Sciences at Boehringer Ingelheim, and Shelley Beluski, who is the Executive Director of Non-Clinical and Bioanalytical Sciences at Xencor. Now let's start with some background. At a very high level, the IRA can be considered a process that tracks a company's holistic position on immunogenic risk. Next, performing an IRA is an EMA FDA expectation which may be utilized by other health authorities. And the approach for conducting an IRA is described in the 2017 EMA and 2019 FDA immunogenicity testing guidances. Next, our survey results suggested that the IRA may or may not be submitted during the IND CTA stage. We have also seen that the IRA practice has matured in many companies. So the IRA may be maintained as a living document or review process incorporating manufacturing changes, patient populations, additional clinical indications, etc. We have also learned that the IRA provides internal documentation of the discussions through the different phases of preclinical and clinical development. And then finally, the IRA serves as a component of the integrated summary of immunogenicity, which is typically submitted at the BLA, MAA stage or equivalent. Next, let's go over some history of the IRA. The practice of performing an IRA began over 10 years ago, with many companies following the recommendation described in the 2014 FDA guidance on immunogenicity assessments for therapeutic protein products. And for many of us, we use this framework that I show you here in this table to collect and rate information using the set of product and target patient-related factors, which are off to the left. Also, many of us in the early days may have developed a risk matrix tool, which looks like the one that I'm showing you here, and defining risk factors as either low, medium, or high, with the goal to define an empirical overall risk for a given molecule. And as we'll discuss, this has often been considered challenging to define risk in this fairly simplistic way. However, the practice of performing IRAs seems to be evolving in many companies, which may be driven in part due to the development of more complex modalities. And the use of a more complicated framework, as shown here versus the one I showed you in the previous slide, may be considered useful to try to further refine the risk. And here is another example of a risk assessment provided by one of our team members that applies a similar approach as for CMC PQAs, with the goal to hone in on risk by focusing on probability of developing ADA versus the potential consequences of ADA. And this example highlights nicely the interest some companies have in trying to understand the risk for immunogenicity using all possible available information at the time. Now let me go into the highlights of our IRA survey, which was performed in 2022. The survey consisted of 25 questions, and our sample size for respondents included 19 companies, mostly large pharma over 10,000 employees, whose portfolio predominantly include monoclonal antibodies, but also a broad set of modalities, including multi-domains, fusion proteins, peptides, oligo cell and gene therapy, enzyme replacement, and viruses. So to summarize the categories of questions that the survey included, we broke it down into a couple of categories, including what I mentioned, company size and therapeutic modalities, as well as the IRA practices, application of preclinical risk assessment tools, clinical immunogenicity bioanalytical strategy, and regulatory feedback. For the remainder of the webinar, I'd like to go over some highlights from the survey. First, we have learned that most companies have indeed adopted the FDA EMA recommendations to conduct IRAs and to prepare ISIs. We've learned, too, there's an interest in improved in silico and in vitro tools to help with predictivity and, importantly, try to drive early decision making. 3, we're going to discuss the limited value in non-clinical ADA evaluations and toxicology studies, especially for MABs, and also the interest to limit or even eliminate this assessment. And 4, there is a strong desire to simplify the immunogenicity testing paradigm, which includes signal to noise in place of titers, particularly for low-risk molecules, developing NAB assays based on risk, and use of surrogate data when appropriate. And then finally, the desire to have some guidance on expectations for understanding impact of ADA on PK. And with that, I'd like to hand the presentation over to Shelley, who will go over some insights for non-clinical immunogenicity assessments that inform the IRA.
Thank you, Christine, and hi, everyone. I appreciate the opportunity to discuss the two main segments of non-clinical immunogenicity assessments, the investigation tools for immunogenicity risk assessment, and the immunogenicity bioanalysis supporting GLP toxicology studies. I'll begin with a brief review of the relevant regulatory guidance recommendations. Most broadly, in 2011, the ICHS-6 R1 stated that sponsors should consider the use of appropriate in vitro alternative methods to animal studies for safety evaluations just in general. In 2014, the FDA's guidance for industry on immunogenicity assessment for therapeutic protein products applied similar language specifically to immunogenicity risk assessment by recommending consideration of in vitro and in silico analyses as a supplement to animal studies. In 2017, EMA's guideline on immunogenicity assessment of therapeutic proteins added more detail in encouraging the use of emerging technologies like novel in silico, in vitro, and in vivo models, with a specific nod to the utility of functional cell-based assays to evaluate innate and adaptive immune responses. So these guidances acknowledge the emerging technologies and encourage their use, but are not prescriptive as the industry investigates multiple methodologies, standardization, and predictive power. This leaves many wondering which tools to use, when, and why. Thanks to Viva Jawa and colleagues that published this figure that breaks down many of the assays by the stage of drug development, starting with the drug discovery phase where many sequences can be assessed in silico for attributes including HLA binding, chemical liabilities, and aggregation in order to optimize the sequences and identify candidate molecules. Then, with the knowledge of the drug's mechanism of action, you can select a suite of in vitro, in vivo, and/or ex vivo models to investigate the steps of antigen processing, presentation, effector cell activation, and ADA generation. To characterize biophysical properties, there are cell-based assays to inform manufacturing process optimization. Finally, there is the integrated clinical PK/PD-ADA dataset to identify clinically relevant immunogenicity through causes and risk mitigation strategies, such as HLA haplotype exclusion criteria and next-generation molecule optimization. This survey queried IQ member companies on the current use of some of these in silico and in vitro tools. First, we learned that many of the IQ companies are utilizing IRA tools in their workflows, and that they are commonly used to differentiate a lead molecule from otherwise seemingly equivalent candidates. Only 2 responding companies were not using in silico analyses to identify peptide sequences that have potential for binding MHC. Because the survey did not link information on company modality or mechanism of action to these responses, we can't determine why they are not using in silico tools. Of those that reported using in silico tools, we sought to understand which databases they were using. These questions allowed the 19 responding IQ members to select multiple answers. For those not familiar with this technology, there are many databases available, and an ideal database must contain extensive, high-quality data on known immune responses to effectively predict potential immunogenic regions within a biotherapeutic sequence. Not surprisingly, the free immune epitope database was reported to be used by 9 companies, and commercially available predictive software was used by 6 companies. Proprietary custom programs were also used by 10 companies, and the use of the humanness scores was mentioned in the other comments. With regards to in vitro tools, only 3 companies reported using the dendritic cell activation internalization assay that detects upregulation of co-stimulatory molecules and cytokine secretion, regardless of antigen processing and presentation, such as binding to innate immune receptors, Fc receptors, danger-associated molecule patterns, or DAMPs, or aggregates. Moving downstream to the assays that measure the ability of a processed and MHC-presented peptide to activate T cells, 12 companies reported the use of the typically PBMC-based CD4 T cell activation assay, and 11 companies reported the use of the T cell dendritic cell co-culture assay. Both assays may report CD4 T cell proliferation activation markers like CD25 or cytokine production. 9 companies reported using MHC-associated peptide proteomics, or more commonly known as MAPS. This is a mass spectrometry-based technique that identifies the specific peptides from a drug that are presented on MHC class II molecules. As noted in the other comments and working group discussions, the therapeutic mechanism of action is key in the selection of these tools. Since the development of this survey, more details have been published by other working groups regarding the use of various innovative tools for immunogenicity risk assessment prior to clinical development. This working group is aligned with the European Immunogenicity Platform and the AAPS Immunogenicity Working Group in recommending harmonization of the IRA tools to enable comparative data analysis, more extensive reporting of case studies linking relevant immunogenicity with non-clinical IRA data, and with an ultimate goal of increasing the confidence of sponsors and regulators of the most value-added methodologies to reliably inform their drug development. Now I'll transition to the immunogenicity bioanalysis supporting GLP toxicology studies with a brief review of the relevant regulatory guidance recommendations. Starting again with the 2011 ICHS 6R1, it explicitly states that ADA in animals is not predictive of a potential for antibody formation in humans. ADA data should be characterized in repeat dose studies to aid in the interpretation of other observations and findings. In other words, ADA data are investigative. And ADA data should not drive a decision for early termination of a study unless it compromises the interpretation of the study. In 2014, the FDA guidance cites ICH 6, but meters the statement of utility in stating that animal studies are generally limited in their ability to predict incidence of human immune responses to a therapeutic protein product, but they may be useful in describing the consequences of ADA. FDA also indicates that the sponsor may consider the use of mouse models to assess immunogenicity. Then in 2017, the EMA splits the difference by stating that the predictivity of animal studies for evaluation of immunogenicity in humans is considered low. Furthermore, they indicate that in vitro or in vivo studies aiming at predicting immunogenicity in humans are not normally required. They also indicate that it is preferable to optimize the manufacturing process to remove impurities and contaminants than to establish a preclinical testing program for their qualification. Finally, this is the only guidance that indicates that the immunogenicity assay should be validated for use in repeated dose toxicity studies. With these subtle differences in guidance, we were interested to learn how non-clinical immunogenicity data were being used by the IQ Consortium member companies and whether more consensus is emerging. First, we determined that most sponsors include non-clinical data in the immunogenicity risk assessment. Amongst the working group, we determined that the decision to use non-clinical data may depend on the drug class, integration of several non-clinical data sets, such as the in vivo, in silico, and in vitro data previously discussed. Or perhaps there are just other factors not assessed in this survey, but this appears to be an area where we lack consensus practice at this time. Despite decades of immunogenicity assay development and validation experience, questions persist regarding immunogenicity assay validation and testing strategies for non-clinical studies. As previously mentioned, only the EMA guidance explicitly recommends validation of non-clinical ADA assays. This working group is aligned with the EIP in recommending that a fit-for-purpose validation, otherwise known as a qualification, for non-clinical studies, since the intended use in non-clinical studies is to use ADA data in an investigation of study observations and findings. While our working group is philosophically aligned, the results of our survey indicate that the current practice may not have adapted. Only 4 respondents said ADA is not routinely assessed in all GLP toxicity studies. As per the guidance, some companies only perform ADA for repeated dose or longer duration studies. While not covered in this survey, one potential reason for performing ADA in single-dose toxicity studies can be to characterize the PK when standalone PK studies are not performed, like in cynomolgus monkeys. When the respondents do assess ADA in GLP toxicity studies, most are validating rather than qualifying the assays. This is likely due to ensure global acceptance and alignment with the current EMA guidance, and the survey did not differentiate between validation and fit-for-purpose validation, nor did it explicitly define the components in a qualification versus a validation, so there may have been some ambiguity in responding to these questions. Finally, recent publications have supported an emerging trend to streamline non-clinical testing strategies by applying a screening cut point based on 99% confidence interval without confirmatory or titer assays. In practice, 18 responding companies are performing the screening assay, 11 companies are performing confirmatory, and 7 are performing titer. It appears that the streamlined testing strategy is emerging in current practice of these responding IQ member companies. However, the extent of adoption may be underestimated since these responses were not linked to the modality or mechanism of action to differentiate those that are maintaining the historical standard from those that have unique scientific rationale. For example, one may confirm when there is a known matrix interference factor, or if they may proceed to titering for vaccine pharmacodynamic endpoints. More investigation may be warranted to identify the roadblocks in implementing the fit-for-purpose validation and screening-only test strategy that is proposed by EBF and endorsed by this IQ working group. Where we have gained a great deal of consensus is in making neutralizing antibody testing rare in toxicity studies. 17 out of the 19 companies do not routinely characterize NAB in toxicity studies. Rational exceptions include high-risk molecules such as endogenous counterparts, novel modalities such as gene therapy, where they are pre-screening AAV NAB in monkeys. In summary, the results of this survey have led this working group to make the following recommendations. Harmonize the immunogenicity risk assessment methodologies to generate standardized datasets to enable comparative studies and establish a knowledge database for the industry to apply to creating protein therapeutics with lower immunogenicity risk. Include the non-clinical data in the IRA, but be clear in the assay capabilities and its limitations. Streamline the non-clinical ADA fit-for-purpose validation and testing strategies as per the recommendation from the EIP. Thank you for your attention, and now I'll hand the microphone over to Kelly Coble to cover the clinical immunogenicity assessments portion of the presentation.
Thank you, Shelley. Hello, everyone. My name is Kelly Coble, and I'm very happy to be here today to share with you the clinical immunogenicity assessments and to share with you really 4 main points. The first point is additional ADA characterization beyond titer, the use of signal to noise, assessing impact of ADA and PK assays, and the integrated PK/PD ADA data-driven approaches to support decisions on the use of NAB assays. So first, let's talk about clinical ADA characterization. On the left-hand side of this slide, you can see a flow chart that describes ADA sample analysis that we're all very familiar with. First, samples go through a screening assay. Those samples that are found screened positive then go into further characterization through the confirmatory assay, and then confirmed positive samples can then be further characterized in assays such as the titer, the neutralizing antibody, or even more specialized characterization assays such as domain specificity, epitope mapping, and cross-reactivity. During the working group discussions on clinical ADA characterizations, we aligned on the following 2 points. First, limiting additional ADA characterization for low-risk molecules beyond titer. And second, we acknowledge that complex biotherapeutics and those higher-risk molecules, including those with non-redundant endogenous counterparts, multi-domain or fusion proteins, peptides, oligos, cell and gene therapies may require additional ADA characterization. For higher risk molecules performing post-study follow-up, immunogenicity monitoring may be needed, but we did acknowledge that recommendations are still emerging on how to interpret the until ADAs return to baseline from the ADA guidances. For complex biotherapeutics, additional characterization of immunogenicity may be valuable, and this can be including domain specificity, epitope mapping, isotyping, cross-reactivity, or IgE assays to aid in the understanding of impact of ADA. Now transitioning to perspectives on use of signal-to-noise for clinical ADA reporting. We did note that signal-to-noise is a growing practice, but use of the tier approach still seems to be the current process. We did also observe that practices are evolving on the use of signal-to-noise, and it may be more applicable for assessment of impact on PK rather than impact assessments on safety and efficacy. As this is a very dynamic area, let's just stay tuned to see basically the next steps. Health authorities have also recommended submitting both titer and signal-to-noise during presentation at conferences, and we did acknowledge that signal-to-noise offers operational ease, resource efficiency, reduced sample volume, though it may not lower sample analysis fees. it was noted that dynamic range, hook effect, drug interference, and precision must be assessed during method development and validation to support the use of signal-to-noise as a titer alternative. And it's encouraged to have early discussion with health authorities on clinical plans for when both titer and signal-to-noise are needed and when signal-to-noise alone can be used. We did also discuss the complexities on how companies transition from titer to signal-to-noise with ongoing projects, and that more discussion and sharing of experiences in this space is very much needed, and that also discussions with health authorities on the use of signal-to-noise in product labels and educating healthcare professionals on the use of signal-to-noise data with regards to clinical impact is needed. So no transitioning to ADA impact on clinical PK assays. What we took away from the survey and shared in the paper is that there is a disconnect between health authority expectations and guidances around the assessment of ADA impact on clinical assays. Our results showed that most companies either perform the assessment proactively or upon request, and it is generally understood that a positive ADA control is only a surrogate and may not be reflective of ADA in patient samples. We did observe a lot of variability among companies on how the assessment is done, and we did describe some approaches that were shared, such as spiking ADA positive control into various levels of QC, parallelism, and dilution linearity, using dilution of ADA positive clinical samples, spiking ADA positive samples with known levels of drug standard, acid dissociation techniques, as well as correlation analysis have been used. And now on to clinical NAB assays and some essential highlights. We noted that health authorities expect clinical immunogenicity strategies to include evaluation of neutralizing capacity of ADA during clinical development, and that the majority of the companies surveyed do not routinely perform NAB analysis prior to pivotal trials, do not routinely evaluate magnitude of response by titer and NAB assays, and discussions with the health authorities integrated clinical PK, PD, and ADA datasets may supplant a validated NAB assay. So in summary, our recommendations for clinical immunogenicity assessments. First, limit additional ADA characterization for low-risk molecules beyond titer, and consider additional characterization beyond titer for complex biotherapeutics and those higher risk molecules. The use of signal-to-noise is growing, but more work is needed on the use of signal-to-noise on impact assessments beyond PK and transitions of ongoing projects from titer to signal-to-noise. Opportunity exists for alignment on how the assessment of ADA impact on clinical PK assays should be evaluated, and in discussions with health authorities, integrated clinical PK, PD, and ADA datasets may supplant a validated NAB assay. And with that, I turn it over to Christine to talk about the integrated summary of immunogenicity. Thank you very much.
For this final section of the webinar, I will quickly touch upon the integrated summary of immunogenicity with regards to industry practices and perspectives that we learned from this survey. We only asked a single question on the ISI, and here are some of the takeaways. The ISI typically contains detailed information on the immunogenicity profile, as well as its impact on PK/PD, safety, and efficacy. And the overarching goal here is to make a case for approval. The ISI is becoming a common practice for BLAs and supplemental BLA submissions. As we mentioned, it's required by the FDA and the EMA, but global health authorities will also consider the contents of the ISI. The ISI provides an opportunity to gather some feedback at the pre-BLA, supplemental BLA stages, and also may enable dialogue and written responses from the health authorities, which really may help get some alignment between the companies and health authorities. And then finally, the working group believes that a well-written ISI may reduce regulatory questions during the review process. Finally, here are some recommendations from the working group for the IRA-ISI process. First, we believe that standardizing an IRA template may add value. I realize this might be challenging to do, but the reason we're proposing this is that there may be some gaps for companies that don't have the expertise. Conversely, Some companies may benefit from simplifying. Some, for instance, may be employing a risk matrix that's too complicated. Second, we believe that some additional guidance beyond the 2014 FDA document would benefit us from helping assign risk and putting risk in better context. So for instance, companies struggle with what is low, medium, or high risk. So considering some common nomenclature could be of value. Finally, we propose the advantages of moving away from the concept that the IRA, ISI be living documents. Maintaining these documents as a process is a lot of work. So what we suggest is to utilize the IRA throughout the drug development process and for life cycle management, and then use the ISI for approvals, and that's when you would incorporate clinical data and the statistical analysis. So this concludes the presentation. I'd like to once again acknowledge the IRA Working Group, which I had the privilege of co-chairing with Sue Richards, who recently retired from Sanofi. We certainly had a lot of fun and many provocative discussions on the IRA process. I would also like to acknowledge the entire IQ Immunogenicity Working Group, who was led by Ben Wu from Genentech, as well as Sue. And finally, we thank the IQ organization for supporting our working group and giving us the opportunity to learn more about industry IRA practices. Thank you all for attending, and now we are happy to start the panel Q&A discussion.
We thank you for attending. I invite the panelists to come off of mute and put on their cameras, and we'll start the Q&A. We've already gotten a few questions, so I just want to remind everyone that they can type their questions in the Q&A box. That's the little box in the towards the right that has the question mark, and we have until 12:30 Eastern time, so we'll try to get to as many as we can. I'd like to start. This is an interesting question because we sort of made it sound like the IRA was sort of something you could choose to do or not. So the first question is the IRA not mandatory for IND submissions? So which one of you would like to take that first? Oh, Sue.
Yeah, I'll jump in. So, I think the expectation is that the regulators expect the IRA information to be provided at the time of IND. And most companies, as we learned in the survey, do actually greater than 90% do immunogenicity risk assessments. The ones that don't, likely they felt that in the comments section, they're dealing with very low risk monoclonal antibodies. So that being said, the question is more of when you provide the immunogenicity risk assessment formal document. And that's where we got a lot of different responses. And the percentages of the responses actually are in the publication. So if you have the opportunity to look at the paper that was published early this year, one of the figures shows the breakout of how companies responded on the immunogenicity risk assessment document and its intent. And it was actually a smaller percentage that said that they include the formal document with the IND submission, but majority provide the information at the time of the IND submission in some other aspect within the document. So the answer is yes, there is an expectation, but you don't necessarily, there is not the expectation that you have to submit your formal IRA document. The information assessment should be done and the information should be included.
Thanks, Sue. And I'd like to follow up. Where would you put it in your IND? Or would you put it in your IB? So how have you all been handling the risk assessment piece, especially for your first submission?
I can start on that. In my experience, we've not been asked for a formal document, but the information, as Sue said, that we would be asked particular questions, would come back if we didn't have it somewhere. Where we have typically put it in the common technical document would be in the pharmacokinetics written summary at the very end. There's a putting an immunogenicity risk assessment and bioanalytical plan in there. Can be a quick blurb or a couple paragraphs, something like that. That has helped prevent too many detailed questions or, directives of like, you must have an immunogenicity assay or something like that for the clinical study. Yeah. And then in the IB, that a very, very small blurb on testing strategy. But yeah, I found the IB, in my experience, it didn't seem to fit in very well. But then you get questions if you don't get it in there.
Thank you. So then let's move to the next question. This is from Alyssa Oquendo. Are companies including their in silico in vitro data in IRA ISIs? Which I think is a very important question, because I think a lot of companies generate this data, but it's not clear, whether to share this or not. And then the second part to this question, do you see any value in including these data in the IRA ISI shared to the health authorities at the time of the IND? So let's just start with putting it in the IRA ISI in vitro assays in silico, and then we can take the next question about sharing it. So for the internal documents, I can start. In my past, we have, I can't say that we've done it for every molecule, especially in silico, but I think it is helpful, especially if you believe your molecule is low risk, to provide that in silico assessment and maybe not rate it as something that has, signifies risk or not, just provide it as information.
Yeah, if I can add to that, I totally agree. It'd be useful to have the information there in your IRA document. The question also becomes how much of the detail you put there versus an overall summary, and then note that, because usually you'll get, especially if you outsource the work, you'll get a pretty detailed report back. And so that gets us back to the concept of the risk assessment also being a process, that you have like a repository place where you collect and keep all the information. So it's always available in kind of a one-stop location. So if a question does come up around a particular HLA type or some in vitro assay, one can go back and actually have the details available without having to put a lot of extra information in a formal IRA document if you use that process.
Kelly or Shelley, do you have anything to add?
Not really more from me, but I do think that the data is very valuable to share and to include. But I think also too, it really has to be put into a holistic picture of what does it mean and what do we understand from it, and how do we understand that data in the context of what we're actually observing during the clinical development of the molecule. So I think it's an important part of the story, but it's also has to be acknowledged as just a singular part of the story. It needs to be put into context.
Yeah, I absolutely agree with all of those responses. And I guess the one additional point I have is that I actually have had a regulator ask for it. So
Wow.
that was surprising, but it's happened now. And so, and again, it's just more information. And as we've talked about, some of the areas that really need to happen are kind of standardizing our methodologies and our databases, and really coming to understand or define what database you use and the meaning of that information and its strengths and limitations. But putting them in the right context and letting them know what you did learn and what you couldn't learn from it seems to be satisfying for them to see some evidence of whether it is or isn't going to be immunogenic in people.
Yeah, that's a really good point, because companies don't always go back and reassess how informative, and I don't want to use the word predictive, because there's many aspects to a molecule being immunogenic aside from the in silico and some of the in vitro data. But to see how it helps inform once we get into the clinic, because we don't always have that information at the time of the IND submission. So at the time of IND, the utility tends to be a lot more for candidate selection and de-immunization and some of those aspects. But it's not clear, and I don't think we asked the question quite that way, is how many do companies go back and reassess that data in retrospect with what they're seeing clinically in their phase 3s, for example.
Which would be a really good point. And then the second half is about putting the in silico and in vitro in the IRA and ISI. And in the conversations I've had with many folks across the industry, is this is more used for internal decision making. And I don't know if there's great comfort in always sharing this. So any of you, please share your thoughts on your experiences with actually sharing these non-regulated, non-required assays in your submissions when you include the IRA and the ISI.
In my case, I've included it as just kind of the summary of the whole body of information. Like, we also have this information that informs us in this way, and then there's also other components. So you've got your tox study in animals, you've got your in vitro, your in silico. It's all a bundle of information that contributes to the case for why you think it's safe to put in humans and may not have any problems, or may, and then we're monitoring certain things.
And this is also similar for my company as well. So I don't think it's provided in a great level of detail, but kind of as a portion of a greater story or a narrative in which we're trying to support basically the continuance of the molecule into the clinic.
Great, thanks. Next question. Okay, given the increasing trend of accelerated development for biotherapeutics using phase 1/2 data, do health authorities expect NAB assays to be established earlier in clinical development when such data are pivotal for approval? That's a really good question. And I think some of this also gets murky when you're dealing with oncology trials where you don't really know when your pivotal trial is. And I know we use that term a lot about not using NAB for pivotal. We didn't really define what pivotal is. So who wants to start first with that question?
I can maybe start just quickly, Christine. So in our experience, yeah, we are always kind of, positioning to try to have the NAB assay for, maybe to low to a moderate risk, biotherapeutic ready for, pivotal, which is what we assume to be phase 3. But there's always cases in which it could maybe be earlier than that. And so this is why we make a practice of retaining and banking NAB samples in earlier trials. And then that then allows us, once the NAB assay is available, to go back and use these samples and analyze them in the NAB assay and be able to collect that data when we go into having interactions with the health authorities. Hopefully then we'd be able to provide that data from some of those earlier trials, if in fact, basically those then become identified as being pivotal.
I can add to that too. So we also bank, so we've got the samples available for later implementation, but we're also using the totality of the data. So we've got PK, PD, ADA. We can integrate that entire data set, and sometimes a PD assay is completely revealing the mechanism of action, and if you see your loss of PD, then you've demonstrated neutralization that's at a clinically relevant level. So there's that. And then we also tend to use free PK assays for our throughout the study as the universal one. Sometimes we'll do a total to investigate. But that means that if we end up having particularly a neutralizing antibody, that's going to disrupt our ability to measure free PK. So if we start to see a loss of exposure, we can then dig in deeper and do a total assay and see if we've actually got a neutralizing that's just blocking binding and the drug's still there, or if drug's getting cleared or that type of thing, to differentiate why we've lost the signal in PK. But the true standalone neutralizing antibody may not always be necessary.
Yeah, we discussed that very vigorously. At least amongst our working group, there was a fair bit of consensus on NABs, in our experience, have some limited utility. It remains to be determined how the health authorities perceive the industry position, but I think we were all getting more better versed in how to use all the other data to supplement the identity of a neutralizing ADA response. Sue, did you want to add anything?
Yeah, I mean, I agree with all the comments. I would only highlight an exception area. is when you're dealing with a high-risk replacement protein. There you're into phase 1/2s, and you're often in the patient population already with phase 1/2s, and then going into a phase 3, especially if they're lifelong therapies. So in those cases, I mean, you would want to, for the phase 1, to bank samples until you have the assay ready, but you may want to definitely consider starting to develop your assay already in support of the phase 1/2 trials for replacement proteins.
Thanks. Next question. When in vitro immunogenicity assays exist, they generally are poor predictors of clinical immunogenicity. Do we really think this work is still needed for drugs that do not target the immune system? I'm assuming that gets back to the T cell, DC T cell activation, those kinds of assays. And I think it's a really good question. We've discussed this, like some of those co-culture assays, for instance, they don't really fully recapitulate an immune system. So, I can start answering this question. I don't think we typically do these kinds of assessments for low-risk molecules or molecules that don't target the immune system. There's usually got to be a reason, I feel, to do them. I don't know. In our group, I didn't get the sense that companies were doing this for all comers, doing these kinds of assessments.
I think also they're selecting which ones to use. It's not like they do the whole battery of them.
Right.
And then also, I think we're in a position where so many different methodologies, and like I mentioned on the databases, you've got many different databases with a variety of types of, well, the breadth and depth of the data set varies between them. So when you lump it all together and say it's a poor predictor, we're making that assessment based on lack of consistency in the methodologies that are being lumped together for analysis. So I don't know if we fully understand yet the predictability aspect, that we actually have initiatives ongoing in the industry where there is the desire to standardize how the methods are being used, and be able to start combining the data sets from various companies, various molecules, and better make a comparison of whether they're predicting properly. And then, as Sue mentioned earlier, actually going back and revisiting after you get to later stages, or and be able to compare it back to when you did those tools, did it really predict? So we need the database that puts all of that information together to really understand how informative is it when the conditions of the assays and the technologies are standardized.
Thank you. Kelly or Sue, do you want to add anything?
I don't think I have anything additional to add on that.
I think what I've seen, just to add, and it basically complements already what's been mentioned, is I've seen many people will use the in silico first tier because it's pretty relatively easier to use, then move over to the MAPS assay because the in silico sometimes can be over predictive. So depending upon your compound, move over to the MAPS assay, and then on a more as needed basis to the cellular assays because of the nature of the complexity and how you do the interpretation of the data and so forth. So they could be tiered to an extent if one feels should do them. So what's any input, Shelley, on those?
Yeah, I was thinking through that. So what I found is a combination of kind of moving through the steps of processing an antigen, and then also considering the complexity of the assay. So like the MAPS is very complicated. So not everybody has those technologies. And I find people tend to start a little earlier. They'll do the in silico, like you said, because that's the first quick and somewhat easier. The bioinformaticists might not agree, but do that first. But that only tells you what epitopes could possibly bind an MHC. Then you need to take it to one of the assays that actually tests cells that are able to process and present, and see if a cell really does take and process and present that epitope. it can be a little quicker and easier to do an activation assay and see if those activate cells and lead to downstream cytokines or something. So, so moving progressively through it and testing those things that can ultimately stimulate the whole process. And then if you want to go down and do the MAPS process, then you're actually sequencing the peptides, and you can go back and address the peptide presence in your sequence in the engineering.
Yeah, and this question is also more specific. Do we really think this work is needed for drugs that do not target the immune system? And I think risk isn't just based on the targeting modality, because again, as Sue pointed out, replacement therapies are high risk and they don't necessarily target the immune system. So it's really more about your modality and its inherent risk, not its risk in targeting the immune system. Why don't we move to the next question? And this is a good one too. Can you elaborate more on whether sponsors need to include in vitro prediction data for IND pack? I think we kind of answered this, but not so much the prediction. It was more about some of the assays. In my opinion, I don't feel like we're at a stage where we can reliably predict. So I would say, no, I don't think the sponsor needs to include their prediction, their in vitro prediction. But what do you all think?
I guess I can add, at least from our perspective currently, I would say we don't either. And I think it's in a large part because I don't necessarily know that we're all, at least internally, comfortable with our prediction at this point. So I would say that we do not include our prediction as part of our submission documents.
I can say the same. I haven't seen them in the documents that have been submitted previously for an IND package of having that specific data included.
Okay.
And I will just kind of say that the same thing back earlier, that when we do that work, we put it together and kind of summarize very briefly the totality of the data that we have that leads to our bioanalytical strategy. So we are taking a leaner approach because we have these data that suggest that it may or may not be that immunogenic. And yeah, is it lean or is it a more strategic, broader bioanalytical package, more complex? And yeah, it is just supporting it with the totality of the data that we have, but not with putting too much weight on any one item, just the totality.
And then the next question, I don't know that any of us could answer because we're not the right group. What is the percentage of biotherapeutics with ADA responses? What is the percent subject showing NAb? So I can tell you from the other group, which I presented earlier, and that group looked at 93, took information from companies over 93 molecules. Then every single molecule that was submitted to that survey had an ADA response. So I would, conservatively say that pretty much all biotherapeutics everyone will see some sort of an ADA response. It could be quite low. And then out of that, how much is neutralizing? Again, it depends on your ADA response. But, I think it's very hard for us to bracket a subset. I don't know if you guys want to opine on this one.
Yeah, I'd like to add that, by design, the assays that we implement are very sensitive assays, and that's by, in order to meet the regulatory guidance of and the technologies that we use now, they're very, very sensitive assays. So really, we're looking to see what is the total antibody that develops against a protein, and all novel proteins all proteins have the ability to mount an immune response. It's whether or not they do and in which populations. So, so when you talk about the incidence or the prevalence of an immune response, it's based on a highly sensitive antibody assay that's going to tell you about every possible antibody we can detect out there. The important thing is then going downstream and seeing whether it really has a clinical impact. Do you see an effect on your PK? Do you see an effect on the PD? Is there an adverse event associated? And that starts to tell you where your clinically relevant immunogenicity is.
Yeah, but I think, a lot of us discuss that NABs often miss providing any more useful information, but it's very hit or miss. So I would encourage the person who asked that question to look at the other working group's publication. And if you, for whatever reason, can't find the manuscript, please reach out to me and I'll send it to you.
If I can add, Christine, on that
Yeah.
question. So there is a lot of information in, as you said, the other publication.
Yeah.
And one thing that it did show, and there's a very nice bar graph in there, that the incidence, there's ADA that's detected across all modalities, to your point, Shelley. And they do have one that shows the incidence numbers, and they're less, obviously, for monoclonal antibodies and more for other modalities, like much higher for, say, the replacement proteins. But it is interesting that across all modalities, there are examples given of a greater than 50% incidence. So, why
Yeah.
the biology, is it something intrinsic in the molecule? Is it overly sensitive assays? I mean, there's many factors, but it did show a really nice and interesting breakout of that type thing. And again, the pieces about monoclonal, the NAbs, the challenge there is if you're also dealing with a competitive inhibition assay, a ligand binding assay, or a cell-based assay, you again get into the sensitivity issues of, nanogram, microgram per mil versus only the microgram per mil for the cell-based assays. So it ends up being complex.
I mean, I think, and that's exactly it. I think this is why the NAbs can be not very informative, because it's technically very challenging to make them useful in the first place. And a lot of us feel, using the totality of the data, you can pretty much deduce that you have NAbs. So, and again, I don't know if the percentage is as important as looking at the impact. This is another NAb question. Are there any special recommendations concerning requirement of NAb assays for single dose treatments? I haven't worked on many single dose treatments, so I'll open it up to you three to see if you have any experience here. I mean, if that is your only dose regimen, I suppose you'd have to do it.
So in our particular instance, we have one that can come to my mind really quickly. It is a single dose treatment, not chronic administration in any way, shape, or form. And the overall, cellular mechanism is incredibly complex, and it wasn't something that was able to really be a neutralizing antibody assay was just incredibly difficult, if not almost impossible to put together. And so the approach that we use in that particular case is that we really assessed the ADA to the molecule, and what we did find, it was also a smaller molecule and a much quick half-life after just a single administration, so it didn't have a lot residence time. But the point is that we were able to utilize the ADA data, and the reality is that we didn't have any ADA data that was positive, right? So we didn't see any positive results in this particular case.
You wouldn't do a NAb.
And so then there was no need for a NAb. However, there was a lot of discussion up front in the preparations for basically, with the regulators, as we were being very transparent, is that the reality is that a neutralizing antibody assay would be very, very difficult to near impossible to basically pull together. And so we were planning then to use, PD as well as the ADA results and, these type of more of a composite data to really help us basically with that narrative in the instance that we were to have seen any ADA, which we didn't, but
yeah,
we did.
Yeah, and I think a special case, I don't know if the participant is alluding to AAVs and the NAbs for that. I think that is a special case. And one of the things that we didn't touch upon too much in our survey is anything too heavy into gene therapy. And I think that is one example of a single dose therapy where you would, and then there's NAbs and TAbs and all that, but that's not something that we vigorously discuss. I don't know if Shelley or Sue, you want to add anything to a single dose treatment?
The only thing I would add to a single dose treatment is if you're giving one administration, to think of it also in terms of exposure. Because like you mentioned in your example, Kelly, it had a short half-life. It basically wasn't present to be presented and processed and so on. Versus you can have other drug modalities that, yes, it's one single administration, but it's in circulation for days to weeks. that's a whole different thing because you have the opportunity to be presented then to the immune system. So I would also suggest thinking in terms of exposure to the individual, in addition to treatment and how many doses.
You took my answer, Sue, so
Oh, okay.
well done.
Okay, next question. Any insight on whether proteinaceous visible particles and their in vitro immunogenicity potential is evaluated? And how about high molecular weight species? So these are very CMC specific questions, which we didn't really touch upon.
It's in the diagram that around the time we broke on the presentation. But yeah, some people apparently are. I can't say that I have experience in this yet, but some people are applying some of the in vitro assays to assess innate stimulation from high molecular weight species or different components of process contaminants, or there's a better word for that that's not coming to me, but different components that they can isolate from the manufacturing process and assess the characteristics of those. So some people are doing that, but I personally haven't seen it done a lot.
Yeah, I remember being part of discussions and looking at some of the publications. There are a few companies, as you pointed out, Shelley, that are doing it and have published some nice papers, and it is mostly innate, looking at cytokine responses. To some of these features. And the high molecular weight species, I think that's also been discussed in some papers, and there's this feeling that you have to have an enormously high amount of high molecular weight species to get anything to be picked up in these in vitro assays. So we haven't seen evidence that any of these in vitro immunogenicity assays are particularly useful for bracketing out some of the CMC features that could pose an immunogenic risk. All right, why don't we move to the next question? Given various publications which demonstrate congruence between signal to noise and titer, what are the perceived hurdles to uptake of wider use of signal to noise and use of titer across companies, given the time and cost savings which could be realized? And I think this is something that we have a new working group, by the way, under IQ that we're tackling as to how to, really implement this, and when, and what are companies looking at. But I'd like the panelists to weigh in, because this is certainly an area that's hot and somewhat controversial.
I'd like to just add one comment about the publications. There are several publications, as is pointed out at this point in time, but we should be cognizant of and try to expand the learnings there to other drug classes, because they tend to be monoclonals in general of what gets presented in the publications, as well as a couple fusion proteins. So those entities will have an FC component, and they may also have more restrictive epitope density than would other type of modalities. So that's something we seem that we need more experience in. Not that they should not be used for low risk monoclonal antibodies, but to try to expand it broadly to all biotherapeutics, we need more experience with other modalities. And then also keep in mind that when we're looking at and comparing titers and time points over time, the population of antibody that's at 6 months and the population of antibody at 1 year, even though the titer may be the same or within one dilution, which is no change, the population of antibodies present may be different specificities, different affinities, and how does that all impact reporting to signal to noise? And so there, as we get into trying to understand its utility in other types of modalities with more broader epitope regions, I think we would get a lot of insights also.
I think one thing I noticed in the data set that we published, there was talk about how do you manage the conversion between doing titer and signal to noise. And I think, right now there's a prevalence of felt, you know, how do you, comparing both when you do your BLA or something like that. But I think there's an element of time since we started really using or looking at signal to noise as an option instead of titer. It's the time of, since we've been doing that and the progression of a molecule through drug development. So I feel like that adoption rate may evolve as time goes on, and those newer drugs in development make it to their IND or their BLA and NDA and all that. So I think maybe we're at this point seeing the transition and maybe not so much adoption like at the early phase. So it'd be interesting to see at phase 1 who's using what technologies and how are they doing the transition.
Yeah, and I think we talked about that a lot. We were a lot of our conversations were focused on how do you use your current data and transition that to signal to noise. But I think, Shelley, I think it's a great point about the evolution that's going on, and some companies are thinking to start fresh with signal to noise, which may be easier than trying to take your already generated data and repackaging that. I think the efforts that have been done thus far have validated that signal to noise in lieu of titers can be accomplished. It can be not only informative, it may even be better in some cases, but you know, there's also, we talked about the caveats of transitioning. A lot of companies that are very interested in this are sharing both sets of data with the health authorities. You know, even though our survey was done in 2022, at the time, no company that participated in our survey had actually submitted signal to noise in the absence of the traditional 3-tiered. So it is definitely something that's evolving.
Yeah.
Why don't we move to the next question, because we have less than 10 minutes. On the topic of in silico in vitro assessments, I think we've kind of covered this. They're not required for an IRA. You know, is there a minimal data package? You know, I think we've all concluded that any in silico in vitro is not required. It's something that's typically done by companies for internal decision-making purposes. The next one is in vitro, a predictive accuracy let me read the question. Is the predictive accuracy of the in silico tool dependent on the modality? For example, could you use it for oligos? Oligos are their own special case, and there's not usually a distinct epitope. It's more of a structure. But I think that is one of the challenges, that they're really designed for proteins and not necessarily, you know, we've had these discussions certainly with some of the vendors that have these packages about synthetic peptides, for instance. And I think some of the in silico databases are getting better at broadening out to some of these non-traditional modalities that aren't protein-based. But, you know, again, the predictive accuracy, I think this has been the holy grail. We're all struggling with anything that has predictive accuracy. I want to go to a question from Amy Rosenberg. She writes, as caution, need to target critical information to limit what health authority reviewers need to evaluate immunogenicity risk and include methodologies like in silico in vitro appendices that can be referred to. So I think I don't want to paraphrase Amy, and I'll follow up with you later, that to be careful what we're sharing, and we fully agree. Let's see. Oh, this is a good one. Is there a requirement for how often the IRA should be updated with emerging clinical immunogenicity data during clinical development? And we definitely touched on this. I think in the early stages, when all of us were adopting the 2014 recommendation, I think there was this feeling that we were perhaps updating too often. And one of our collective learnings was that you can really back off. But, you know, I'd like for the panel to chime in on when they're updating their IRAs, especially, you know, in the later stages of clinical development.
So maybe I can start. For us at Boehringer, I think what we do is we have a kind of a fit for purpose, right? So in earlier development, we're adding in the data that we have, and we're having that high level discussion, but acknowledging that there's still a lot of components that will come with later development. I would say as our molecules move into further development and maybe even closer to submission, obviously this document, there's more time spent on it, there's more data that's put into it. It's definitely something that is a higher point, a higher priority for us to make sure that we complete in a very thorough way, and try to understand also the narrative by which we're understanding, you know, immunogenicity and the impact of immunogenicity on our molecule. So it does live, you know, when it starts with the molecule, you know, as the molecule is developed, but definitely the document becomes a lot more complete and thorough the closer that we get then to submission, if that helps.
Yeah, I'll add a little bit there. I mean, there are certain milestones in clinical development that you may want to consider then revisiting the document. So clearly, if you're adding in a patient population, moving over into pediatrics, if you have process changes in manufacturing, is a logical place for it to get revisited. In terms of incorporating all the clinical immunogenicity data, keep in mind that'll also be showing up in all the clinical study reports. So you don't have to repeat by trying to put that all into a immunogenicity summary. So again, it gets back to how the ISI is used and how your IRA document is used. So if your ISI ends up being as is proposed, your document for your submission, for your eCTD submission, then you would not necessarily have in there including all phase 1 or early documents. You may align that with the safety database patient population, for example. So it kind of depends, but there's other places that data lives. And that's why it also, what we were talking about, a process. If you have a place where information is shared, that you have your immunogenicity section, for example, in your project team's SharePoint, you'll have access to those documents. Or you can have a section that pulls in the clinical study report. So whoever is the immunogenicity lead, whether it be that point in time or in the future, because people do change roles, the information will be all available in one spot for a person who then picks up in a later stage on a project.
Thanks, Sue. So we have 3 questions. One is quick. We will be sharing the slides. So Amy, you'll be seeing them. The other, one of the last 2 questions is when to do these predictive assays, the MAPS, the T cell activation, the DC T cell assay. I think a lot of us discussed that the main value is doing candidate selection. There's not so much value unless you want to diagnose your molecule and why you're seeing higher than expected immunogenicity to do these later, so you avoid ever doing this again. But I think the common practice is to do it as early as possible to try to root out any potential problematic candidates, with the caveat that we still don't know how predictive these are, and there's always this risk of potentially excluding a molecule that could actually be very important, you know, in the clinic. And, you know, it remains to be seen if they would have been truly immunogenic. So the final question we can try to tackle is a short one. How do we justify pre-existing antibodies? You know, I'm not quite sure the context here. I've always seen pre-existing antibodies not as a nuisance, to say the very least, from an assay perspective. Kelly and I have had a lot of conversations, and so did our working group, about what does it mean when you see it, especially for a particular modalities such as nanobodies, single chain FEs. Some just have these epitopes that the immune system cross-reacts to from recognizing some naturally occurring antigen. There have been a few cases, very rare, where there's been consequence of pre-existing antibodies. So I don't think it's something we ignore. But, you know, I'd like for the panel in the last 2 minutes to share their thoughts on how they justify pre-existing antibodies and going forward.
I think one of the keys is making sure that you know what the pre-existing drug exposure is. So, you know, we all buy procured samples, and we're hoping that they're not exposed, because we ask for them not to be exposed to large molecules or whatever type of molecule you may have that could cross-react with your molecule. And we hope that's true, but you don't really know till you get in study, and you got to know what your inclusion exclusion criteria are. So there's a chance of prior exposure, like if you're doing an antibody, if somebody else has seen an antibody and there's some part that's shared, that you can see that. So that's one justification. But as Christine mentioned, also just innate immune responses that exist from environmental antigens or other prior infections, that type of thing could be pre-existing and cross-react with your molecule. The other thing to consider is how does that impact downstream, and there is a risk that when you dose that it could boost the immune response. So you may see an early onset of ADA. So it could be impactful.
Absolutely. And maybe I can add, just from some experiences we've had internally, is that with some of the more highly engineered molecules, we have seen a kind of a resurgence in some ways of the pre-existing reactivity, and how I mean, it is a characteristic of the molecule, it is, we deal with it, we have to assess it, and we sometimes will even spend, if it's concern, we will make an effort to be able to distinguish following administration what is happening to the pre-existing versus what is happening to the treatment emergent portion. And so that's one piece, but I think it's also could be a valuable portion too. We've used it to help understand the epitope by which basically has been, created, that is driving this. And then this basically identification is then used for next generation molecules to try to engineer that epitope out, because it was of course unintentionally done so. And so I think pre-existing is something that does happen. I think you have to pay attention to it. And I think it also could be informative for engineering efforts later, for future molecules as well.
It also can be that we're introducing it early on as one of our in vitro assays that we assess early on in a molecule to help determine if they've created something in our pool of candidate molecules that cross-reacts, and let's eliminate that from our candidate selection.
Yep, we do the same as well, Shelley. So it's also becomes a tool for us for helping to identify candidates as well.
Yep.
Yeah, and to add, it's trying to understand, as you've heard from Shelley and Kelly, what is the reactivity to? Because if you think about pegylated molecules, for example, there's so much PEG in products these days that the prevalence in the normal population has increased to anti-PEG reactivity. And then if you think as of another example of pre-existing reactivity with cetuximab, I mean, there the antibody was an IgE response. Patients who had never seen the drug before had an anaphylactic type response, and the antigen was determined. Actually, it's an interesting story because it was more geographical, and it was because of tick bites. So it was an environmental allergen that the individuals made an antibody response to that now cross-reacted with an epitope on your drug. So their cross-reactivity of some sort or another, if it's truly a baseline person, but what the cross-reactivity is to, whether it's a environmental exposure or if it's to previous shared epitope with some other drug, is an interesting problem to kind of sort out.
Thanks. I don't know if we have time we're a little over time to squeeze in one more question. This is the last one, but I think it's a good one. And we may have to think about this because we can, and I also want to add that I am answering the questions and taking them off, so the responses will be available. How does your ISI being used for health authority, I think it's reevaluation, if you have a combination of two molecules? We've discussed this. Like, do you write separate ISIs for each of your two, or do you write an ISI for a combination? It sounds like it should be a working group. It's a biggie. Have you ever done a combination for two? Anyone on the panel?
I have not.
I'm about to, so I'd love for somebody to recommend.
Well, I think we could take it up with our working group when we try to streamline how to do that. But that is a very good question that I don't think, I know we've discussed this internally, and you're going to have separate documents for each molecule. Combining it, I'm sure it is a special case, and that is to be continued.
Yeah.
With that, I want to
seems like you'd have to talk about it some. Yeah. Go ahead.
I want to thank everybody for joining. The slides will be available. The presentation will be on YouTube. I want to thank the panel. You guys did an excellent job, and I want to thank our working group. So with that, I think we will close out this webinar. Thanks again.
So long, everyone.
Thank you, everybody.
Have a great day.